All right, chapter 14, moving right along. So today is a really interesting and often controversial topic about biotechnology and genomics. So this is looking at how technology has influenced science for the good and again controversial. So as technology has advanced, there's been many things in science that has also advanced that has allowed us to use science to influence medicine, but also like the food industry, things like that. So that's what we're getting into: DNA technology, biotechnology products, gene therapy, and genomics. So biotechnology is the use of natural biological systems to create a product or achieve some other end desired by humans. So this is not normal, but achieving something that is beneficial to us, something that we have thought, this would be helpful for us, whether it is a crop that produces more usable food, or it is a medical procedure that is helpful to humans. These are the kinds of things that we are looking at. And with the advances in technology today, we are able to do more of these kinds of things. It allows scientists to modify specifically the DNA and the genetics and the genome and change them up and tweak them here or there to make them a little more user friendly, so to speak, and make it so that we are able to manipulate them in a way that it works a little bit better for us. So it can be to improving a characteristic, make a little more biotechnology product, genetically modified organism, GMOs. We've heard a lot about that, especially in the food market. More and more you're hearing about non-GMOs, so food that does not have genetically modified organisms that are being used. But a GMO is an organism that has one whose genome has been modified in some way, usually by using a recombinant DNA technology, which we'll get into in a little bit. A transgenic organism is an example of a GMO that has had a gene from another species inserted into its genome. So now we're kind of playing mix and match with the genetics and the DNA, taking DNA from one organism, putting it into another organism. This kind of brings into mind the idea of cloning. Now, cloning is having two organisms that are exactly the same. It's important to note that this takes place naturally in the natural world. Plants clone. If you cut a little sprig off of an aloe plant and grow that, that's cloning. If you have a sponge plant and there's a piece of that sponge that breaks off and starts another sponge, that's cloning. If you have identical twins that came from the same egg and the same sperm, that's cloning. So cloning is something that happens naturally in the natural world, and it's a natural process, but it can be made into an artificial process too. So sometimes cloning can be something that can be done artificially if humans choose to do so. Sometimes it is an easy process if it's something simple like a plant or an animal that isn't very complicated, but sometimes it can be more complex like cloning a mammal. And the reason, of course, that humans are so interested in cloning mammals is because we are mammals ourselves. So if we can clone something like a sheep, like has been done with Dolly, then potentially we could clone a human. And of course, that's the question: is can we clone humans? And if we can, should we? So gene cloning is the production of many identical copies of the same gene. So now we're not looking at a whole individual being cloned, but maybe just cloning a specific gene. So clone genes have many research purposes. If the gene is diseased, well, let's clone the diseased gene in a laboratory setting so that maybe we can do some experiments on it to maybe test that it can be changed and maybe modified so that we can test it in the lab and see if we can find some cures. This also leads to possibility of humans can be treated with this gene therapy. Now, there's also something called recombinant DNA, or rDNA. This contains DNA from two or more different sources. So when you take DNA and combine it together, with something else, maybe another kind of DNA, or maybe from another animal or from another human. When you're recombining that DNA, it's called recombinant DNA or rDNA. This usually requires a vector of something and also some enzymes. Now, a vector is often in the form of some kind of bacteria. Bacteria are just really good in these situations because they don't have a nucleus that you have to deal with, and bacteria often have very fast generational cycles, so you can have see results very quickly from one generation to the next because their generational cycles are very quick. So if you can introduce an rDNA into the host cell, they don't have those nucleus, so they only have plasmids, which are small accessory rings of DNA. And so that's one of the reasons why they are a very good common vector. So 2 enzymes are required to introduce this foreign DNA into the vector DNA. These restriction enzymes and DNA ligase, remember anything ending in ASE is a type of enzyme. So DNA ligase is going to come in and kind of seal up that DNA and opening created by the restriction enzymes that are in that area. This just kind of shows how human or cloning of a human gene takes place. You take the human gene and you take that plasmid you've got from the bacteria, which is the vector again, and you're going to get the gene that you want off of the human chromosome and you insert it into that vector, and then you put it back into the bacteria. The bacteria acts as kind of a host, as a place to grow and nurture that gene until a replication is made and you're able to eventually take out that gene because it's been replicated using the vector of the bacteria. Now, restriction enzymes are a little bit different. They cut the DNA at specific points. They tell the DNA, okay, we need to make a cut here, we need to make a cut there. Cleaving the DNA makes the DNA fragments ending in short sticky segments, and so here we've got like the sticky ends where you can see they were together up here, but they've been cut with that restriction enzyme, so they've got these kind of open places where other materials can come and bind to them. So then you have DNA ligase, and DNA ligase will come in and seal. Remember, ASE is a type of enzyme. So the DNA ligase is going to kind of come and seal together and kind of zip everything back up. And the recombinant DNA molecules have been prepared by this point. And the treated bacteria take up these plasmids. The plasmids are the circular DNA. Bacteriids and plasmids are, as a whole, are going to reproduce, again, those short generational lifespans. And so this is going to make many copies of those foreign genes and allow us to use those genes as needed. So this is going to amplify that we've took that one tiny piece of that DNA sequence and we've multiplied it many times. And the amount of DNA doubles with each replication of the cycle, so we can get that DNA strand from that gene, and we can take it out once it has replicated. So here's kind of our polymerase chain reaction, so PCR. So we can see it taking place and making copies of that segment that we wanted. Just kind of a picture format. So there's also something called DNA fingerprinting. So just like everybody has fingerprints that help to identify them as who they are, our DNA is very unique to each individual as well. So as technology has gotten more and more advanced, DNA typing has become more advanced also, and we are able to look at a person's DNA and determine who they are based on their DNA signature, so to speak. Just like we can look at the fingerprints left behind, we can look at the DNA left behind to figure out who did what. So this is something that is helpful with like forensics and but not only that, helping like figuring out paternal and who's the parent, those kinds of things. PCR amplifies the short tandem repeats, which just stands for STR sequence, which are short sequences of DNA that are repeated many times in a row. So it's kind of like just little chunks that are constantly repeated. And these are found in specific locations in the genome. And the DNA fragments are fluorescently labeled to kind of make them just stand up stand out a little bit more, so they're easier to spot. And then a detector kind of just records these fluorescent areas to help us know where they're at. And we can kind of compare them and contrast them to known DNA samples. And that way, if there are a lot of similarities, we are able to determine, yes, this came from the same individual, or no, it didn't. Each person has his or her own unique printout. And if you have a match, then you can indicate how likely of a match it was and how positive you are that that individual was responsible for leaving that DNA behind. The FBI uses 13 STR locations on various chromosomes for identification of individuals within the United States. 13 doesn't sound like a whole lot, but the 13 can be quite helpful. Short tandem repeats, again, that's what STR stands for. Those 13, when combined together, the more that match, the better and more certain the FBI are. This kind of shows how it works. Here's the crime scene evidence. And you could have the person, the subjects, subject's not the right word. The person who was injured, their DNA mixed in here too. And then suspect number 1 and suspect number 2. And so you can compare who has more matches, and whoever has more matches is more likely going to be the suspect. So you can see that suspect B has more matches, and so these two are more the same, so this one is more likely going to be the one that has more evidence against them. So they're more likely going to be the one that actually was at the crime scene. Now there's also something called genome editing. With genome editing, targets specific sequences in the DNA for removal or replacement. So with this, we can actually take chunks out or add chunks in, depending on what we are trying to edit. CRISPR, it sounds kind of funny, but it's clustered, regularly interspaced, short palindromic repeats. But CRISPR is the most widely used method for genome editing. So taking chunks out, putting chunks in. It was first discovered in prokaryotic, remember, no nucleus, where it acts as a form of immune defense against invading viruses. Viruses are going to insert their DNA into the host cells, causing the cells to form new viruses. That's what basically happens when you get a virus. A virus overtakes your body and puts the virus into your own cells. So it takes over the cells of your body. A bacteria just kind of like lives there and takes over the surrounding area, but a virus actually infects your own cells. So the virus is going to insert its DNA into the host cells. The CRISPR actually system utilizes an endonuclease enzyme called Cas9. Cas9 identifies specific sequences of nucleotides and DNA of invading virus using a guide RNA molecule, complementary base pairs of the genomic DNA strand. So it kind of mimics it and uses the complementary base pairs of the DNA strand to kind of trick it into saying, hey, give me what I need because I've got the same base pairing that you would have normally had. Cas9 breaks both DNA strands and inactivates the virus. So this CRISPR system, the scientists have kind of taken advantage of it, saying, hey, if it can use these chunks of DNA, then why can't we use it as well? And it's used by researchers to target specific sequences of nucleotides and say, we want to study this sequence. Let's insert this here to capture and kind of cut out this sequence, and we can use that for research purposes. This can be used in almost any organism for editing and removing these different sequences. The target is known as a complementary RNA strand and can be used by the Cas9 to produce a break in the DNA. So it basically says we're going to cut the DNA here and here's how we're going to do it. Acts as a form of molecular scissors, so to speak. Shows how the process works, and we basically just cut out what we need, and then we can rely on the cell to kind of fill in the gaps with that. GMOs, we already mentioned, genetically engineered organisms. This is a very controversial topic, but honestly, there's a lot to think about. If you were a farmer who was trying to grow corn and you had a stalk of corn, one stalk of corn that grew 3 heads of corn, or you had a stalk of corn that took up the same amount of space but it grew 10 heads of corn, it's obvious which one you would choose. You would choose the one that grew 10 heads of corn because you grew more corn for the same amount of space. But you're able to grow more and therefore produce more and therefore get more money out of the same amount of space. So if you could genetically modify your crop to produce more corn, it would make sense that you would do so. Watermelon, another great example. People who have watermelon nowadays, they have those little yellow seeds in them that you can swallow. But in the past, we used to have big black seeds, and those big black seeds you don't really see anymore because watermelons have been genetically modified to no longer produce those black seeds. Honestly, it's kind of ridiculous because what kind of plant would ever produce and spend all this energy to make a fruit that they can't use? Because the whole point of a watermelon fruit was for animals to eat it, to eat the seeds, and then carry the seeds around and disperse them and poop them out somewhere else later on. But now that there aren't any seeds, there's no reason for a plant to make such a complex watermelon anymore. So organisms that have had a foreign gene inserted into them are called a transgenic organism. They have some kind of foreign genetic material. A gene of interest is inserted into the bacteria. We already mentioned this. The bacteria is allowed to grow. It feeds and nourishes that genetic information that was put into the plasmid. And the products in the market include insulin, hepatitis B, vaccine, TPA, and the human growth hormone, clotting factor 8. So all of these are made by genetically modifying bacteria. Bioremediation is the process that uses transgenic microorganisms or other organisms such as plants to detoxify and degrade environmental pollutants. We can kind of make a mess of the environment. It's kind of what we do. So if we can fix that a little bit by using oil-eating bacteria or creating something that fixes a problem, then we're going to try and do that. So one strain was given genes to clean up toxins and a suicide gene to self-destruct after the job. So it was, in order to prevent things from going out of control, let's give it a gene that kind of makes it destroy itself once that process is reached. Genetically modified plants, we talked about this. If you have a crop that can grow more in the same amount of space, that's good. Oh, sorry. Human hormones. Plants are being engineered to produce human proteins, including hormones, clotting factors, and antibodies in their seeds. Because if we can get those benefits from the food that we eat, why not give it a try? This is showing a genetically modified plant. Apples, when they were cut before, they used to brown very quickly when they were exposed to the air. Now they brown much more slowly because children especially, they see that brown, they used to think, oh, that's gross. But now, if it's browning much more slowly, it's not as likely to seem as gross, and children will eat them, be more willing to eat them. So we can insert genetics or those genes into the eggs of an animal. This is called microinjection and also vortex mixing. So sometimes the eggs are placed in an agitator with DNA and silicone carbon needles. The needles kind of puncture the egg so that the DNA can get into it. The agitator just mixes everything up. The needles make tiny holes through which the DNA can enter. Fertilized eggs develop into the transgenic animals. This has been known to introduce the gene for bovine growth hormone to eggs for the purpose of producing larger fishes, cows, pigs, sheep, and rabbits. So again, making it so that we change things just a little, and hopefully it'll be enough. Gene farming is the use of transgenic farm animals to produce pharmaceuticals. if we can get an animal to take the medicine and take the risk for us, and that way we don't have to take the risk, we're all about that. Some people are against animal experimentation, and that's totally fine. That's up to you. But if we can use the animal and animal benefits to help a human, a lot of people are going to prefer that. Protein appears in the animal's milk. The technique has been used to create drugs to treat cystic fibrosis, cancer, and blood diseases. Just showing how the process works, we can take a gene out of a person or a mammal and inject it into an egg, and then we put that into the sheep. The sheep is, and the milk has put into the host mother. The host mother had some babies. Of those babies, one of them became identical, and that was, we put our donor egg in there, and now we can see we have the mother that was eventually cloned. So Dolly was the first sheep to ever be cloned. Dolly did die young, but we don't know if Dolly died because of the fact that she was cloned or because she did catch a genetic disease. So the first one that was cloned was in 1997, was Dolly. So they took an egg from an adult female, and they took the nucleus out, and then they took a stem cell from an adult female, and they switched the nuclei, and they zapped it with electricity to get it started, and they put it implanted into another female. The female then accepted it and carried that baby to term and gave birth, but then another female was the one that actually raised Dolly. So it was kind of a three-mother process, one to provide the egg or the information for the egg, one to provide the nursing, and one to provide the one to provide the clone, one to provide the egg, and one to provide the nursing. So kind of a three-step process. Animal cloning is a difficult process with a low success rate, usually 1 or 2 viable embryos per 100. So development it's a very complicated process. There's not a lot of success. It has been done, but we don't know if the disease that Dolly died from was a natural process or was related to her being a clone. So there's also climate change and overfishing has led to a decline in wild-caught salmon. Researchers have developed an alternative transgenic salmon called the Aqua Advantage salmon. 99.9% Atlantic salmon with gene production from the other 2 fish. Growth hormone, gene promoter, wild Atlantic salmon and the Aqua Advantage salmon are the same size when they reach adulthood. So they're trying to figure out ways to save the animals that we currently have. For many years, researchers believed the adult vertebrate animals would not be cloned. Then in 1997, they did do it. They're still cloning mostly young and mostly from babies. They can't clone just a regular adult, and part of that is because of the just difficulties of raising an adult, and it's just easier to have a young be born and not have any or not raise an adult in the lab, and it's easier just to develop a baby and give birth to a baby and that kind of thing. It's also very, very expensive to do. Sometimes there's the idea of therapeutic cloning where you just clone a specific organ that you need. So like if somebody is going through liver failure, maybe you just clone the liver because that's all you would need, or maybe you just clone the hand because they lost a hand. Maybe you just clone a heart because they're having heart failure. So climate change and overfishing had led to a decline in wild-caught salmon. Researchers have developed... Oh, we already did that. Okay, here we go. So this shows the two different types of salmon. This shows the promoter gene and the gene segments that are mixed into the population, creating kind of a different kind of salmon there. Gene therapy has the potential to cure many diseases. There are ex vivo, which are outside the body, and in vivo, which are inside the body gene therapy. So some ex vivo, ADA and SCID. One of the earliest uses of ex vivo was for familial hypercholesterolemia. In vivo, cystofibrosis, nasal respiratory spray, liposomes, and lentiviral vectors. So those are all in vivo or inside. Ex vivo is gene therapy. You've got your defective gene. You're bringing in some bacterial with the correct DNA to try and fix that broken gene and trying to restore it. Genomics is the study of genes on humans and other organisms. The Human Genome Project is producing a working draft of all the base pairs in all the chromosomes. It took 13 years to sequence 3 billion base pairs along the length of the chromosome. The project involved universities and private labs throughout the world, making many, many different kinds of and making this genome project. So it's been a huge work of progress. I believe they've got it all mapped now. All the genetic information of an individual or species, that's kind of the point of it. Goal of the Human Genome Project is to determine the base pair sequence and construct a map showing the sequence of the genomes of a specific chromosome. Humans have between 19 to 21,000 genes. Most code for proteins. We learned about that in one of our previous chapters. Much of the human genome was formally described as junk, but it's not necessarily the case. It does not specify the order of amino acids. RNA molecules can have a regulatory effect. Polymorphisms were identified. Most polymorphisms vary by only one nucleotide. Structural genomics, knowing the base sequence, is being followed by functional genomics. Historically, genes were defined as discrete units of heredity that correspond to a locus on a chromosome. Eukaryotic chromosomes are much more complex. We already knew that because eukaryotic cells in general are more complex. Showing kind of the introns and the exons, we already removed that. Introns are removed, exons stay, and then the exons get spliced together. Intergenic sequence are DNA sequences that occur between the genes. Repetitive DNA elements, 2 or more nucleotides repeat. And transponsons are specific DNA sequences that have the remarkable ability to move within the chromosome sequence. So they kind of move around within that sequence. Unique non-coding DNA is the majority of this intergenic sequence. So exons, they're the ones that stay. Introns are the ones that are removed, repetitive and unique. Transponsons, specific DNA sequences that have the remarkable ability to move within and between chromosomes. So they're kind of more like gene regulators, kind of regulating what's going on within that gene. And the ALU repetitive element. They were described 60 years ago as the jumping gene, and he was identified in corn. And they've been discovered in fruit flies, bacteria, and humans. So here's a purple one, here's a yellow one. Modern definition of the gene should remove the emphasis from the chromosome and place it on the result of transcription. Remember transcription versus translation. Transcription is moving to mRNA. RNAs are also a useful product. Genes need to not be on one locus on a chromosome. DNA sequence can result in one or more products. Genetic material need not be DNA. DNA microwaves can monitor the expression of thousands of genes simultaneously and tell us what genes are turned on, environmental conditions that turn on the gene. So different things can turn on the gene, and microwaves arrays of the DNA help with that. Contain microscopic amounts of known DNA fixed into a small glass slide or silicon chip in known locations. Genetic profiling, called the person's genetic profiling, may indicate if a genetic illness are likely and what type of therapy might be most appropriate. So they're not always foolproof. During pregnancy, they ask if you want to find out, if you want to do a blood test to find out if your baby has any genetic diseases. You can accept or decline. It's up to the mother. And it will only tell you if the baby is at high risk. It won't tell you if the baby really has that genetic disorder. It'll just tell you if it's at high risk for it. If it is at high risk for it, you can choose whether to go on to do more invasive testing. All right, comparative genomes compares the human genome to the genomes of other animals. We are very closely related to the chimpanzee. We only have a couple genes that are different. Model organisms have genetic mechanisms and cellular pathways in common with humans. Scientists inserted a human gene associated with early onset Parkinson's disease into Drosophila. Flies showed symptoms early to humans. Flies might be able, instead of mice, to study therapies for early onset Parkinson's disease. That's helpful because flies have a faster generational time than mice do. Just a different chart showing organisms used to study external genome and external of the genes. DNA probe, DNA probe, and tag DNA did not probe. Proteomics is the study of structure, function, or the interaction of cellular proteins. Entire collection of a species' proteins is called a proteome. Proteome is larger than a genome. Understanding the protein function is essential. So making sure you understand the overall shapes that their body can make and the special training that they understanding the protein functions is essential to the development of better drugs. Correlate drug treatment. The particular genome increases efficiency and decrease side effects once the primary structure of a protein is known. Bioinformatics is the application of computer technologies, and homologous genes are genes that code for the same protein. So sometimes we see the same protein making a repeating pattern. So even though it codes for it has different genetic coding, it's going to show the same protein when it is all spelled out in an amino acid. Metagenomics is the study of metagenomes, genetic material obtained directly from the environmental samples. Seeing if there's anything else. I'm going a little fast here. I want to just read the slides. I'm trying to pick up the information that I feel is important. This has to do a lot with computers. Eukaryotic DNA can be identified and removed, leaving the microbial DNA behind for analysis. Cut the DNA of entire chromosomes into small fragments and clone. Sequence each segment and arrange based on overlapping nucleotide sequence. This is showing them in a pond, kind of collecting some of that material. And if we research different areas, we can use this to discover more about our natural world. All right, so we're going to go ahead and stop there. Stay tuned for our next chapter. And if you have questions, I know I sped up a little bit there at the end, but again, I just wanted to make sure we had enough time for everything. You can go back and do some more reading on the slides if you are interested. If you have questions about anything, let me know. Bye, guys.